this video, we will be talking about membrane potential. Membrane potential refers to the difference in electrical charge between the inside and the outside of the neuron. So right here I have circled, which is going to be the plasma or the cell membrane of the neuron. It basically separates the inside of the cell from the outside environment. And then I have written inside and then outside. We'll basically just say it's the inside and the outside of the neuron. The difference in electrical charge develops due to the grouping of ions on the inside. So, inside and outside of the membrane. So, right here I have drawn in different colors, circles. Those are going to be our ions. And ions are basically atoms that have either lost or gained electrons and thus have a positive or negative charge. So, there are several ions that play an important role in the membrane potential of neurons. There are positively charged sodium ions, which I have here in blue circles. Then we also have negatively charged chloride ions that I have drawn here in green. And then basically when a neuron is at rest, the sodium ions and chloride ions are more prevalent outside of the cell. Down here in the yellow, we have positively charged potassium ions, and then various negatively charged ions, often referred to as organic anions, represented here by these gray circles. When a neuron is at rest, the potassium ions and organic anions are more prevalent inside of the cell. So at rest, the inside of the neuron is more negatively charged than the outside, causing the resting membrane potential of an average neuron to be around negative 70 millivolts. So over here, I have drawn a sodium-potassium pump. Basically, one way this potential is maintained is through a mechanism known as here the sodium-potassium pump. So this is a transport of protein that uses energy to constantly pump 3 sodium ions out of the cell, while at the same time we are pumping 2 potassium ions into the cell. And because there are more positive ions being pumped out than in, it helps to keep the membrane potential negative. And unlike other ions, potassium tends to move fairly easy across the cell membrane through what we call ion channels, which are basically membranes spanning proteins that allow ions to pass through. And potassium will pass out of the neuron until it reaches the point where it is at an equilibrium, and when forces like diffusion aren't pushing it at one direction or the other. And so at this point, the membrane potential of the neuron is around negative 65 to negative 70, which is known as the resting membrane potential. So over here in this slide, I have basically the initiation of membrane potential change. So what we call an external stimuli, it's basically a neurotransmitter binding to a receptor, which causes the opening of specific ion channels. And so if the cell depolarizes enough to reach what we call threshold potential, which is around negative 55 millivolts, voltage-gated sodium ion channels open. And so here is where action potential basically happens. So what we call depolarization is Na plus rushes into the cell, making the inside positive relative to the outside, which we see as a positive 30 millivolts. And then once we hit repolarization, voltage-gated K plus channels open, also allowing K plus to exit, which is restoring negativity inside the cell. Now, at hyperpolarization, the cell briefly becomes more negative than the resting potential due to the slowness of the K plus channels closing. And so, returning to the resting potential, the sodium, potassium pump, and ion leach channels restore the resting membrane potential over time. and the process resets the cell, readying it for the next stimulus. And then, so basically right here, I just have a better visual of -70 when this is the initiation of an action potential, and then it charges up all the way to +30, and that's when Na plus enters. Once Na plus enters, we're gonna go back down and -K plus leaves, and then once we're like at -55, that's when we're at a threshold. But we're gonna keep going down to hyperpolarization, and this is when K plus has not yet closed but slowly closing, and then once we're like say in this area back up here, that's when it's fully closed and we are back at -70. So that was the process of membrane potential. I hope that helped, and I hope that was a good understanding.